Click for IB Biology. The principles of respiration. Here we're studying the Krebs cycle, otherwise known as the citric acid cycle. In this, the acetyl group imported from in the link reaction is progressively broken down but through decarboxylation, and how this is linked to the reduction of coenzymes, and in particular to the formation of reduced NAD. We will also see that there's a direct synthesis of ATP within the Krebs cycle. A PDF of this is present in the description below, along with a quiz, so that you can test your understanding. The Krebs cycle is, of course, a cyclic metabolic pathway, and its details were determined by Hans Krebs. His image is shown here. And Hans Krebs was awarded the Nobel Prize for this work in 1953, the same year that Watson and Crick published their structure of DNA. Like any cyclic metabolic pathway, there is an acceptor molecule, and this is the 4-carbon oxaloacetate, the name of which you do need to know for the exam. And this picks up the 2-carbon fragment called the acetyl group. Of course, this is delivered to the pathway as acetyl-CoA. So the CoA drops off the acetyl group, it attaches to the oxaloacetate group and forms the 6-carbon citrate molecule, which is a tricarboxylic acid group. And of course, this is composed of 6 carbons, 4 plus 2, making 6. Then there are the carbons are then broken off this structure in what is called decarboxylation. This will occur twice to give us what is called progressive decarboxylation. So the citrate, 6-carbon, is broken down into a 5-carbon structure with the release of carbon dioxide. The name of the molecule is of no significance here. The important thing is to focus on the decarboxylation of 6-carbon to 5-carbon, the release of carbon dioxide, which of course will be excreted, and also there's the formation of the reduced coenzyme NAD. Later we will link this NAD to the electron chain system and the formation of more ATP in our next video. The third step is what's called regeneration step, in which the 5-carbon compound is then further decarboxylated to a 4-carbon compound with further excretion of carbon dioxide. So notice that we've got 1 carbon dioxide here, a 2nd carbon dioxide here, and that's consistent with the idea of breaking down the acetyl group, which was imported into the Krebs cycle. Further NAD is reduced, and once more this will be transformed into ATP at the electron chain system, and there's also substrate-based ATP synthesis in this final stage. So the classic stages are the acceptor stage, progressive decarboxylation, and regeneration. We will now go on in the next video to explain how the NAD, the reduced form of NAD, can bring about the synthesis of ATP. In this image, we have summarized the steps into the classic cyclic diagram. So briefly, we can see the link reaction, the acetyl-CoA. Step 1 here is the acceptor stage, 4 plus 2 to give the 6-carbon, the citrate, the decarboxylation here, the formation of the reduced coenzyme which will then go off to the electron transport chain. The 5-carbon is now going to get involved in step 3, which is regeneration of the oxaloacetate, and in doing so we remove a carbon here. That's another part of the progressive decarboxylation and further formation of the reduced coenzyme. This last stage also results in the formation directly of ATP. So as previously stated, the next stage to deal with is how the energy which is being put into the form of the reduced NAD, how does this bring about the formation of ATP, the subject of our next set of videos. Links to those videos are appearing below, along with the subscribe button and links back to the playlist so that you can look to other videos in this sequence.